welcome everybody to this, I think is it the last seminar of this semester's Open Science seminar series. We are really happy that we are joined today by Tim Errington, who is Director of Research at the Center for Open Science, that aims to increase openness, integrity, and reproducibility of scientific research. In that position, he conducts and collaborates with researchers and stakeholders across scientific disciplines and organizations on meta-science projects aimed to understand the current research process and evaluate initiatives designed to increase reproducibility and openness of scientific research. Such projects include large-scale reproducibility projects, such as the Reproducibility Project Cancer Biology, a large research project replicating 50 experiments from cancer biology. Very wonderful work, congratulations. And the DARPA-supported Systematizing Confidence in Open Research and Evidence and Evaluation project on new initiatives, such as Open Science Badges, registered reports, and a novel responsible conduct of research training. So thank you very much for joining us. We are really honored to have you here, and you're welcome to start your presentation.
Great, thanks for the invitation. Let me share my screen quick. Everything look all right?
Perfect.
All right, great. All right, well, yeah, today I'm gonna talk to you guys about some thoughts on replicability, reproducibility, and I'm gonna highlight the cancer biology project. I think the timing is good for that since we just published it. So I'll use that a little bit as an example of some of the ways that we think about this. And some disclosure, I think it's always good to do this. We're a nonprofit organization, and below are the different philanthropic groups and government groups that support the work that we do at the Center for Open Science. Okay, so today, what I'm going to walk through is just a little bit of what is replicability, reproducibility, you know, why do we care about it a little bit. I'm going to use the Reproducibility Project in Cancer Biology as a bit of a case study of kind of conducting it and how we assess evidence when we get evidence from replicability. And then a little bit at the end about where do we go, right? What do we do with this? And what can we as individuals do? So there's been, over the past couple of years, lots of emerging definitions around what does reproducibility, replicability mean. I think for the most part, it seems to be landing on kind of this simple matrix. National Academy of Science and here in the United States issued a report that kind of aligns with this, I think, emerging theme. So we can think about, you know, where our data is coming from and the type of analysis code, if you want to throw that in there as well, that's being used, and whether they're the same or different between different experiments or studies. When they're both the same, that's generally what we're referring to as reproductions, and computational reproductions is I think a pretty well understood word for what that means. You know, can I take the same data, apply the same analysis, and get a similar result? That latter part we'll come back to, because I think that gets kind of tricky as we get farther away from this. If I start to collect new data or use different data, right, basically not the same data as the original finding or studies, then we start to get into replicability, or that emerging difference, which is I'm trying to analyze the data the same way, I'm just collecting new data, either a different sample or I'm just trying to redo it in the lab, a different experiment again. Then when I start to change my analyses or change the data, this is where I think we start to shift out into different types of definitions, right? Robustness, when we use the same data but do different analytical techniques. Fields like economics, I think, are doing more and more of that, where they have these very complex data sets and they do a lot of robustness testing to ask, well, is it just contingent on this one analysis approach I did, or if I did different approaches, different assumptions, do I get similar results, different, you know, similar conclusions? And then, of course, if we were to collect different types of data, do different experiments, right, all asking the exact same types of questions, and by definition and extension, doing different analytic approaches, does this generalize, right? Can I draw the same conclusion by having different data analyses all kind of converging on one thing? I'm going to come back to this kind of idea between replicability and generalizability again near the end of the talk. But these are some emerging definitions, and a lot of what I'll talk about today is really kind of in that square bin of replicability, but it applies, I think, across the board in terms of the way we want to think about this, or how I think about it. So when we ask the question, why do we care? I think this is a fine, I don't really love the word reproducibility crisis. I think it's more about, I think that just has a bad connotation to it. I think instead we try to think about, like, is there a reason to be concerned about our reproducibility rate and our practices around reproducibility, I think is the better way to do it. But a couple of years ago, Nature had this a survey done, Wendy Baker there ran it, and asked researchers within their network what they thought. And what you find is that, and I think this is probably still relevant, is that there's something going on. There's a problem, and that we should talk about it. We should do something about it, and it is something worth paying attention to. I think more importantly is who's in this group, right? These almost 1,600 researchers. Who are they? And this one sticks out to me a little bit, right? Which is this kind of question I definitely grappled with in grad school, which is, can you repeat? Can you reproduce someone else's experiment? So I'll use the word repeat here. If you do it again, can you get the same result? And whether it's your own experiment or whether it's someone else's experiment. And what they find is kind of across fields, people say, yeah, I have a hard time reproducing the results of others, including myself, including my prior research. Right? I did an experiment, I tried to do it again, and I can't quite get it to work. And again, like, there's lots of ways to, I think, interpret this. So we can come back to this a little bit of, like, what does this mean to fail to reproduce something or replicate something? There's a lot of different definitions out there, and I think that's actually in some ways not something that we should see as just a simple yes, no, thumbs up, thumbs down. I think it actually has to get a little bit more into what are we looking for and how do we go about trying to find that. And that's really no different than the type of research we do anyways. So let's take a step back even farther from 2016 and think about what other things, especially in the biomedical space, were causing kind of, well, why should we pay attention to this, at least in this current times? And to me, these three papers kind of stick out. One, John Aniti's paper from quite a while ago, kind of, basically it's saying, well, just looking at the findings that we have in research, most are probably false, just the way that we're designing this, right? So it's more of a simulation, just looking at the literature and saying that there's a reason to think that most of those published findings are not what we think they are. And then 2011, 2012, two pharmaceutical companies, Bayer and Amgen, issued their own kind of white papers on having a hard time replicating. They didn't disclose anything here, right? They just said we can't replicate it. We'll tell you our percents, right? 13%, 25% success. But we don't really know anything about the papers that they looked at, what they meant by success. You kind of figure it out a little bit from their writings, but not entirely. So we're still stuck with this question of, like, what does it mean? Why couldn't you replicate it? What did you try? What did you find? What is your definition? But all of these do, I think, point to the question, which is, well, we do know that this matters, right? Like, we do know that the research that we're doing, and say the preclinical space as an example, is a feeder into our clinical sciences. So we do know that we do want replicable science, or at least some level of replicability, and we should be mindful of it to see how we can, increase the ability for us to maximize the return on investment of the research that we're doing. So actually, when these last papers came out is when we started the Reproducibility Project Cancer Biology. So I'm going to use that, and we just published it. I'm going to use that, like I said, as a case study to kind of go through what it means to try to replicate research and what we found, and then, like I said, what this means going forward. So just as a bit of a basis of what this project was, it started in 2013. It's a collaboration between the Center for Open Science and Science Exchange. Science Exchange is a company in California that basically is a marketplace for identifying labs, academic core facilities, or shared resource facilities, and CROs, contract research organizations. So they had a network, they still have their network, that you can basically go and essentially purchase an experiment to be done by people that have expertise in certain techniques. So they were a great partner for us. It meant we had a network that we could take advantage of. And then the other piece that we had was funding. We needed support. That's, again, something that I think is reconsider in terms of steps forward, which is, there's not there was growing, but still not a lot of funding support for replications, as well as for publishing those findings. So we got lucky in the sense that we were able to find a private philanthropic group now they're called Arnold Ventures. They used to be called Laura and John Arnold Foundation, based in Houston, Texas, that supported this project. And then the nonprofit open access publisher eLife that was willing to publish everything that we were doing in here. And again, I think those are two aligned partners that enabled this project to occur. So let me take a step back and think, well, what were we doing? So our approach here was systematic sampling. We weren't looking for specific papers that we wanted to investigate. We were actually just trying to ask this larger question, which is, all right, what is that replicability rate within the literature, and what do we find, and what does this mean in terms of trying to understand replicability and understand what the findings can mean for us as a community. So we started in this 2013, so we looked back, and these are just our decisions, right? Looking back 2010 to 2012, those papers that were published. And we looked at it based on preclinical cancer papers that were making a lot of impact at the time. Impact here being like people who were reading it largely, downloads, citations don't really emerge then. So we were looking at downloads and readers like in Mendeley, those types of databases. And then, like I said, I'm going to talk about this a bit more details. We used eLife as a publisher to kind of work with us on something that I think is very crucial. And I'll come back to this point of really making sure that there's pre-commitment to what we're doing. So what I mean by that is, let's make sure that when we're designing these protocols, when we're about to do a replication, that there's a pre-commitment that it is a confrontation to that original finding. And it's meant to be the best design and a fair test within reason. And then that also means that we have a pre-commitment to ourselves when we do that replication, which is, this is what we said we were going to do, and are we able to commit to that? Are we able to kind of follow through that? So I think you guys had an earlier session on pre-registration, and that's essentially what this is. This is pre-registration, but with replication, pre-registration is really doing a great job of two confrontations: the confrontation to that original finding in the replication, and then the confrontation to yourself, right, in your pre-registration plan. The beauty of it is we also did pre-registration in the format of registered reports. I'm not sure if you guys were able to review this, so I'll go through that. But here we get kind of feedback during that process. So it's not just us saying, this is what I want to do. It's us saying, this is what we want to do, and we'll have it get peer reviewed. And we had to get peer reviewed by the original researchers, reviewers, other experts in the field, and editors, including biostatisticians as well. And then we go through another round of review where we confirm our adherence to those pre-commitments. And the nice thing about here is it'll get published regardless of the result, as long as we follow through on our pre-commitments. And that's an important thing because it's so easy, especially in replications, to want to say, oh, yay, nay, on whether you should publish it based on the outcome. So two big outcomes that we had from this was just the process. We've ran this project for almost 8 years. And there's a lot of things to learn about testing replicability that I'll go through first, and then what did we find, and how do you assess replicability? And I think that's a really fun question without an obvious answer. So just as a review, I'm not sure if you guys went over this. If you guys did go through pre-registration, you're aware of that. But if we think about registry reports, it's kind of, how does pre-registration fit into our traditional publishing model? So if we way oversimplify things and we think of our research as developing ideas, designing this, the study we're going to do, collecting, analyzing data, writing the report, and publishing it, peer review sits way at the end there, which is after I write my report, after I've done all my experiments, presented my results, now I get peer reviewed. And then if I'm lucky and get it through all those reviewers, I publish it. What registry report says is it shifts it into two stages, where there's that first stage of peer review, where after you've developed your idea, maybe have some pilot experiments, design your study, then you have that peer reviewed, and it's peer reviewed on what you're planning to do. So it's talking about, well, what's your design? What's your sample size? What's your statistical plan? So essentially what everything is for pre-registration, but in the peer review process, which is really nice because that means you're getting all this additional feedback as you guys as one's designing their experiments, and in this case, designing our replications. And then the nice thing is when you do that second stage, it's not about what you found, about it being a positive result. It's about, did you follow through? Did the controls work? Did you report it, appropriately? And that's really important, especially in something like replication, where that starts to, there starts to be some tension. This format kind of relaxes that tension in an attempt to reduce the bias that can occur traditionally in peer review. So this is a diagram from a while ago, and it does a nice job actually of describing in a kind of simplistic way of what we were doing throughout this project. And I'll use this because I think it gives us a good roadmap, kind of thinking about replications. So one is starting with the paper, starting with the finding, whether, in our case, it was always someone else's. And I think that's usually a good way to do it. It's independent, but we're identifying kind of those key conclusions from the paper, what data, what figures, what experiments are we most interested in. Especially when you have papers that have dozens and dozens of experiments, I think, you can do them all, but in our case, we chose only a handful, and I think that's traditionally how things are done. Then we started with the paper and started saying, well, let's try to figure out what reagents and materials and methods we can ascertain from the paper to try to figure out what was actually done. Essentially, at this point, we're just reading, right? We're reading the paper, trying to say, do we understand it? And usually what you have, if anyone's done this, I'm sure you all have, which is it's not in the right description. You're always asking questions. You're always trying to say, I don't quite understand what was done. Or I don't have access to the data. I see you presented the data a certain way. Again, I saw you guys had an earlier lecture on data presentation. There's so many ways to present data that really sharing the data enables others to restructure it in any way they want for visualization or analysis purposes. So lots of questions. Then we'd reach out and start filling in those holes, start going back to asking the original authors, can you help us? Can you figure out where this is? Can you figure out where we can get this information from if it's not obvious? And then if we couldn't get information from them or they didn't know, we'd have to just start filling in gaps, making our own assumptions. We'd compile all that, and that's what we'd put in that first publication called a registered report, where we'd send that to eLife, it'd go under peer review, get more comments from hopefully the original authors, but definitely other experts saying, did we get it right? Are these designed the right way to be a fair test of that original finding? And then only after that was accepted, then we'd perform the experiments. So basically we'd follow the protocol, all the reagents, everything that we planned out, we'd follow it until we couldn't follow it. Because definitely things did not go according to plan always. And that's, not unexpected, but sometimes unexpected. And then after we finished all those reports, all those experiments, that's when we'd submit individual replication studies. So we were doing this for a long time, for as many papers as we could. And what we found at the end, this is just going to kind of aggregate kind of our overall findings. So I'll first start with our design stage, right? When we started with all these papers. So we looked at 53 papers, picked out 193 experiments in the end that we were interested in. And what we found is kind of room for improvement. So this graph here kind of illustrates on a handful of different criteria that we were coding outcomes that occurred throughout that entire process. And on the left side, you can kind of basically, as you can see the coding on the left side is kind of that ideal state. On the right side is kind of that less ideal state. So if we look down at the bottom at data sharing, we had very few experiments that had open data, open raw data. And then even when we asked for it, very few authors could share the raw data with us. Over half either didn't respond or did not have data to share with us. And that's a lot. That makes it really hard, right? That makes it really hard to try to design replication experiments or even understand it if you can't have access to the data. All you have is, say, a graph with error bars. So that's similar to analysis as well. Now, this is a little trickier in the sense that not all of these experiments have statistical inference tests associated with them. But even when they did have them, there were many times when we didn't even know what the test was. We just saw p less than 0.05, or even worse, just an asterisk, which you then are saying, oh, I guess that's less than 0.05. And sometimes it's not unexpected because it's very common to have representative images be presented and that's it. So we don't have any variation. And that's, there's good things to that. And then there's challenges that come to that when you replicate it, because now you're left with just a single image, right? A single data point that you have to try to infer from. But a lot of the times we'd have variation, just no statistical test, right? Bar plot with error bars. That's what we'd have. And that's what inference is being drawn from. So this is also a challenge. And this is just trying to think through, like, how do we share more of that data, right? If you're going to show a representative image in the paper, can we see the other ones? That would have been very helpful. We asked, but never really received those at all. Same thing with code sharing. For when they did have analyses reported, it was really only one time that we had access to open code. And then not everyone was able to even share their analyses with us. And we were very liberal in that, just saying, if you have an Excel sheet, whatever it was, can you tell us what test you did so we can understand it? So these are just instant roadblocks to understanding it. if we wanted to do reproductions, right, reanalyzing the data, it's clear that we want to be able to do it for many of these because we don't have the data and we don't have the code. And the data is, you have to have the data in order to do a reproduction, a computational reproduction. So I think this is definite room for improvement. But if we keep going up the ladder and we think about redesigning, right, collecting new data, big parts in the biomedical space is going to be, I need those materials, I need those reagents. And so again, you know, not a lot of those key reagents, animal models, you know, cell lines, antibodies were available openly or commercially. So we had to ask. And we did have a lot of people that were willing to ask. And we'll get to how many actually ended up following through on that. Another barrier is just the hardest one is protocol clarifications, the methodology of the experiments, what was actually being done. And we found, and again, not surprisingly, that all of them needed clarifications. I think the thing that was more surprising was how diverse it was, right? We definitely had a handful of experiments where they were lacking a lot of detail. It's very hard to understand what was done in that original experiment. And when we asked for help, we didn't always receive that help. There were some authors, about a quarter, that were just amazing. And I'll give you some examples of like what that means. But unfortunately, we had about a third that either didn't respond to us or told us to kind of go away. And that's just disheartening. And it also tells us that we have a dependency on having to reach out to authors. And really what we want to do is try to decrease that dependency as much as we can to enable science to kind of be more open, be more reproducible, replicable on its own, not dependent upon individuals, or at least not overly dependent upon them. All right, so what do I mean by all this? So here's just some examples when we look at papers. I'm sure you guys have seen this yourself. So here's a figure that has this key experiment that we want to replicate with a bunch of different findings. Got a bunch of asterisks that say p less than 0.05 or 0.01 or 0.001. Representative images, we have variation and bar plots, but no access to the data. We have a little bit of understanding at least of what tests they were doing, which was really helpful. And then if you go to the methods, they are short and many times also describe going somewhere else, right? We've all seen that, the method trail. Quantifications performed elsewhere, follow that reference to follow that reference to follow that reference, and you've gone down a rabbit hole. And then usually at the end of the rabbit hole, you're not even seeing the method that you were looking for. And that's an instant barrier to understanding what was done, because that means you have to start to figure out what was done. And so then we put together the lists of, well, we don't understand, this is what we pulled from the paper, we don't understand all these questions. Can you help us? Can you help us figure this out? And that's what we'd send to the authors. We'd have a lot of back and forth trying to fill it out. And this is what we were coding at the end that I just showed you. So let me just give you some barriers that we had, right? We wouldn't know what antibodies were used. We didn't know like the sex of the mice that were used in some experiments where it mattered, how long treatment periods were for, what instruments, software were used. We wouldn't know certain protocols, right? They'd say ice stain cells. We wouldn't have any idea how they did that. Didn't even understand how some of the variables were named, right? It'd say positive area. We don't know what that, like, how were you defining positive area? And like I said, data being shared was a barrier as well. You know, and again, like, I don't want this to seem like the issues like, oh, these original authors are the problem. They're not. It's the culture that we have. And I think it's illustrated by just kind of looking at the responses I got over the years, which is it got lost in reducing text size during publication, right? Publications reduce and make it harder for us to be reproducible and replicable in our research, because if there are limitations on words or limitations on the number of figures, it means that it's much harder to do this. We have to figure out ways to get around that, and as well as change that at the publication stage. Some of it is our own practices, right? What are our data management practices in our labs, right? When we do our own research, if we don't have access to those data, if we've lost access to them and somebody else has to ask for them later, that makes it really hard. So the more we can open it up in the beginning, the better off we are. The other thing is that it's distributed. People have moved on, right? A lot of these studies, as you guys all are, probably wherever you are in your career, you work there for as many years as you can to get your degree or until you move off to the next exciting thing. And it means that that lab is left trying to figure out the pieces. And if you haven't communicated as much as possible in the paper and connected to the paper, it's really hard for someone else to try to figure it out afterwards. And again, we had some authors that would do that. They would track down the rest of the people who did those original experiments that have since moved on to their postdoc or since moved on to their own lab around the globe. And that's a lot of work that definitely, I think, are ways to find improvement to make it better. And again, like sometimes it's just not accessible, right? The files are gone or they can't figure out even where to look anymore. So really, to me, it's the problem of how do we incentivize these types of practices from the beginning? All right, what else did we find when we try to conduct replications? Well, we found out that even when we had to keep reducing the number because we had all these challenges of not having enough shared, having to remake reagents, having to spend a lot of time reconstructing protocols, figuring out exactly what types of experiments to do. Even when we could conduct experiments, we realized that not all the reagents that we were going to get shared were shared. So sometimes we'd have to remake reagents, cell lines or antibodies or plasmids. And that experiments, we had to modify them, right? It didn't, like, even though we sat there, had this really rigorous protocol, we're like looking at the original paper, asking the original authors for help, getting it peer reviewed, we still ran into models not working, experiments not working as expected. And sometimes, like I said, it was mundane reasons or relatively mundane reasons, so optimization of the approach. And sometimes it was models not behaving at all like the original study or behaving at all like the intended model. And I think to me, that's actually part of the point of replications, is it also gives us clarity on what are those conditions that are necessary. So I'll come back to that point in a little bit. All right, so let's summarize this. So what did we see? And really the reason to do this is because it lets us think about what we can do to improve replicability. So lack of transparency and accessibility of methodology was definitely a big barrier, as well as sharing, just sharing data, reagents, code. There's a dependency on communication being necessary for some of that information. And we had unexpected challenges. And so that led to challenges in the resource costs. Typically when we think of our research and the funding that's needed to support that research, it's really complicated in the sense that we're not really always saying how much does it cost for each given experiment and how much does it cost to produce a paper or produce results. Here we had a budget from a grant that let us really kind of look at all of this, and all of these resource challenges got more and more complicated as we went along the project and kept on running into these challenges along the way. And so we were able to kind of understand that we weren't able to do as much throughout the project, but we wanted to make sure we kept our quality as high as possible. All right, so we have all these challenges and finally did this experiment, and now we get to this question of, and I think this is what I said in the beginning, right, which is, well, what does it mean to be replicable or reproducible is the same thing, right? How do we assess that? We looked at it 7 ways. There's one more in here that I want to talk about, but we did, and each one of these have disadvantages, some of them more than others, and advantages. But we did want to look at them different ways because there are different ways to assess replicability. Right, just the direction, right? Do I find a finding in the same direction as the original, right? Drug increases survival. If I do it again, do I see an increase in survival? Who cares how big? Do I just see an increase? You can also look at statistically significant, right? You can use the null hypothesis testing framework. We also looked at effect sizes, whether they were in the original was in the replication confidence interval or vice versa. You can also look at prediction intervals, right? What's the replication in the interval that would be predicted based on the original and the replication. We can also compare the effect sizes. To me, that's actually a really good one to think about, which is, well, what's the magnitude of the difference, right? If I see a 10-fold difference in the original finding and I get a 1-fold difference, it's in the same direction, but do I draw the same conclusion? And then you can also do meta-analysis approach, right? Can I combine the evidence to see what I have? There's one more in here that we didn't look at. I didn't put down here, but I think is really important, which is subjective, which is, what do you think, right? What do you as a researcher, how do you assess whether it was replicable or not? In many cases, that's probably part of what was being done in those Bayer and Amgen studies, right? It's probably a combination of some of these and their just own opinion of did it replicate or not. The trick to, I think, all of these is always to try to do them on the front end when you're designing. So we'll come back to that just a little bit longer. But let me first talk through what we were trying to do. So we looked at these 7 approaches and we looked at them across all of our experiments. And most of our experiments were positive results. And so this applies pretty nicely to positive results. And most of them were numerical results. But like I said, sometimes we didn't have numerical results. The original was just a representative image, but we try to replicate it. So it doesn't apply. Can't do this. Right, it doesn't really fit into this very neatly. And we'd sometimes look at original null results. And the reason we look at those is those were still important, right? You'd say the drug increases the efficacy for treating a certain type of model cancer disease, but it also does not show risk to, say, the heart health in the animal. And so there you'd expect to see a null. And so we'd include nulls in ours. It was not that many, but we included them, and that doesn't really fit here either, right? Direction doesn't really make a lot of sense in the null. If you have no, the original says there's no impact, well, nothing's really nothing. So direction in the replication doesn't really have as much weight. So what we ended up having to do is kind of summarize only across certain criteria that could apply to the majority of our findings. And we looked at five dichotomous replication criteria. And so that's listed here. And these are what we find, right? We find that if we are trying to compare if something's replicable from the original to our replication, following that process I just described, that less than half, 47% of them were in the same direction and statistically significant. You know, a quarter of them, the original effect size was in the replication confidence interval. And remember, the replications, when we did them, would be powered at 80% of the original effect size. So our sample sizes were not much larger, but on average, a lot larger than the original as well. We would look to see if the replication was in the original confidence interval. So still less than half, 48%. Again, be mindful of all of these. All of these have caveats, right? If I have a very small sample size, I'll have huge confidence intervals. Pretty easy for me to get within that. We had also looked at the prediction interval. There we had 61%, and then the meta-analysis of the original replication, 63. So wide variability in terms of like how we can think about whether it's successful or not. And it really comes down to what matters, right? What do you want out of it? From our case, we were exploring this. We weren't trying to say for each experiment, what did we want? We were trying to do this systematically and try to tackle this question. So one way we did it, that's an interesting way to think about it, is who cares, right? There's 5, who cares? They're all good. Maybe they're not all good, but hopefully a majority of those are good. So what we did is we kind of looked at those across and said, regardless of which of those criteria, how often did they succeed? And so when we looked at these effects, we had 112 of these that we could do this way. We found that when we combined all of them that we could look at, 46% succeeded on most criteria, so 3 or more. So that's one way to really simplify how replicable the research was at large. And what, like I said, if we look at the nulls versus the positives, you know, we see that they were twice as successful. The nulls were twice as successful than the positive results were on these 5 criteria. Another way to look at this is also just the effect sizes. So these are graphs. We actually modeled this off of a similar project that was conducted a few years ago in psychology, where the approach was modeled off of it for the most part, and then also this graph was modeled off of it to allow some comparability. So what we see here is the original effect size on the x-axis and the replication effect size on the y-axis. The right side here, B, that's just a zoom in of the smaller effects because there's many small effects. And what the color of the dots are whether the replication p-value was statistically significant or not based on a 0.05 threshold. And then we have these density plots for the original up above here, and then the replication on the side. And what we see is that the original effects, by and large, had a lot of positive effects, which we kind of knew, and their effect sizes were larger, much larger than the replications. In fact, on average, we found 85% smaller effect sizes than the replications. And to me, that's actually really, it's something to think a lot about, right? Which is many of the replications were smaller, but the size of that magnitude, 85% smaller, has, I think, huge impacts in terms of the way that we think about the utility of that finding. Also speaks to why is this happening? Publication bias is probably definitely one thing that sits in there. Another thing would be selective reporting, also having a huge impact on this. And small sample size obviously will inflate this at the same time. So what can we conclude here? Well, we find, like I said, that the effects are much weaker than the originals. Depending on how liberal you have it, we have low replicability across it, but it depends on which test you want to use. All those tests are very highly variable across each other. But if we look at them in aggregate, regardless of what the test was, we see that the positive results were half as likely to replicate as the null in terms of, like, 3 or more success criteria. And I think more importantly is that there is room for improvement, right? We're doing amazing things. We ran into a lot of challenges trying to conduct this. And when we found results, we found that, right, like, looks like less than half were replicable. So maybe there's room to improve this, especially the effect sizes. So then we have to get to these harder questions, right? Which is, all right, well, how do we interpret this? And there's more ways to think about this than these three, but these are the three kind of big ones, I think, when we think about when we fail to replicate something, what does it mean? Well, one is the original findings was a false positive. You can go farther and be like, oh, the original was, they made it up, right? I guess that's true. I really hope that's not true in our case, but we do know that there's a small fraction of people that do make up data. So yeah, a failure to replicate sometimes does, sadly, expose things like fraud. But as Johninides was trying to get at, which is it could just be a false positive. It's a statistical fluke, right, that we had it. That could also be driven by the original researchers maybe p-hacking their finding, right? They had a bunch of different analyses approaches they could do. They picked the one that gave them p less than 0.05. It didn't hold up when someone else did it again, because of the variance and the p-value, being very careful of that. Part of that is, how much selective reporting has roles in this as well. Could also be that the replication was a false negative, right? So we got it wrong on the replication. And again, could be statistical chance. There's a possibility for both of these. We tried to minimize them, but it doesn't mean that they're not there. Other parts that could occur in the replication is we did the replication. There's an error in the replication, right? We did it wrong. We designed it wrong. Again, we tried our best to minimize that throughout that entire process, having so much peer review, but we can exclude these as possible ones. But there's another one that's also probably quite true, which is they're both right. Right? They're both right. That the original's right, the replication's right, but right there, right? They're true. But that the difference between the two, again, there's pressures in terms of selective reporting and publication bias. But another big one is that we don't understand the conditions to get them, and that there's differences between those conditions, and that more research is needed to actually appreciate the differences between the experimental design. And in many cases, to me, like this is one of the strengths of replication. It's this really hard question of there are so many variables at play. How do we know what is causing that finding? So this is a paper Brian Nosek, who's our CEO, and I wrote last year, trying to grapple a little bit with this, which is, okay, well, what and how does replication fit into, maturing our theory and maturing our understanding of findings? And really what we got into is replication and generalizability are kind of one and the same, right? That really, since you can never do the same thing again, there's always going to be some variation in that universe of possible things to test, right? That really it's about a question of what would you expect? So if we look on the left here, when we're in early discovery stages, when we don't have a strong theory about what's going on, and that basically means that the potential space of the generalizability of how far a finding could apply to is pretty large, right? In the universe of possibilities. Again, when we think of this, sometimes we write our papers this way, I think, thinking they're a bit bigger than they are, which is, oh, look, I found this finding in a cell line of breast cancer. It applies to every single type of breast cancer in the world. And we know that's not true for many, many, many reasons. One is breast cancer itself is a very complex disease. But it doesn't mean that it's not true, right? It just means you have a huge generalizability space about the way that you want to test it, about the applicability of what types of outcomes and settings it applies to. But over time, and especially as you do replications of that, and then you start to push farther out into, well, let me test and generalize it into different conditions as well. Some people call this conceptual replications. Things that you're going to have different paths. And that's kind of what the right shows, right? Which is if you continually have success, because there's always going to be variation. A replication that succeeds means that you have a better understanding of it. It means despite all those changes that you controlled or didn't control, it still occurred. The finding is still there. And that means you should have more success on it. And it means that it's more replicable. The space that the conditions under which it applies for is more replicable. But the reverse is also true, right? If it fails and you have persistent failure, it means that it's going to keep shrinking. It means that that finding, if it is true, is narrower and narrower and narrower on the conditions. And again, this is something to think really hard about, right? We want to control our conditions as much as possible in our experiments. We know there are many factors at play, but we also don't want to over control them at the same time, right? Because we hope they apply and generalize to the broader biology of the world around us. And kind of the thing we definitely don't want is it to be a technical reason. It can happen sometimes, but sometimes it's kind of the one that I think we're all afraid of, right? Which is, oh my gosh, is it literally that lot number? Was it something funny with the serum and that lot number? And that's the only reason I have this finding. If it is, then that means it's right. It just means that it's only narrow. There's something unique about that lot. And hopefully it's something that tells us more about the biology. but it might not be. And that's part of the reason replication is important. And it's part of the reason why transparency of what we believe the conditions are and the conditions that we did use are important for sharing. And part of this is that you have to have commitments too, right? We have to be able to say kind of beforehand what we believe is necessary to have a successful replication, and as well as even defining the criteria. So again, another way to think about this, and this is kind of what we did in the cancer biology project, but when I think about replications, there's kind of two routes to go. One is, I think sometimes the way it occurs, which is, oh, hey, I've got either my finding or somebody else's finding, and I'm going to replicate something. And sometimes you don't talk to each other. You don't have to, right? A original finder, somebody who did the original experiment and someone who's trying to do it again, you don't have to talk to each other. You should be able to communicate that through our papers and through our methods and approaches. But what we should be thinking about is that confrontation, right? Which is, well, are we doing the same thing? And really what that means is when we get an outcome, can we both agree that the outcome's a fair test? If we don't do that, we take the path to the left, right? Which basically you'll do a replication and you won't really conclude anything because you'll be doing everything post hoc, right? And after you see the findings, you'll say, oh, well, you did the replication wrong. You'll be like, no, I didn't. I did the replication right. And that becomes kind of a little bit in a spinning game where we're just kind of ended up being more in a theoretical debate than actually kind of being grounded more in the evidence. But if we can do pre-commitment, like pre-registration registry reports, then it allows us to be confronting, saying, okay, well, based on the way this is designed and based on the way that we're going to analyze the data and based on what we'd expect, what counts as success or not. We didn't do that, like I said, in our process. But to me, I'd recommend something like this, which is if you want to replicate, go into it from the beginning saying, what do I believe are the necessary criteria to try the experiment again? What conditions do I want? And what outcome counts, right? And you can use the ones that we showed, or you can come up with another one, which can be a lot more simpler, but I do think it's really important to have that pre-commitment, both to what you're designing and then also holding yourself accountable when you do the replication, you see the results. And it's okay, by the way, if it doesn't work out. Most of what you end up with is, great, it looks like there's a difference. I wonder if that's the difference. And that's actually how you generate another hypothesis to do that experiment again. And you keep doing that until you either have realized that it's not worth investing, right? It's not worth going any farther because it's too narrow of a condition, or you realize that that generalizability space is actually more of a replicable space and that you are on the right path and you should keep investing in that line of research. And that's either individually or collectively as a community. All right. So what are some solutions? And again, look through, I think I'm at the end of your pretty awesome series here, and you guys have, I think, seen a lot of these solutions actually in what you were talking about in terms of thinking about our statistical practices. Pre-registration being one example of being something that can help us a lot on this, which is that pre-commitment to what we're doing, specifying what you want to do, thinking through the best way to design your experiments before you do those. Larger sample sizes is something else that comes along for that, right? I think especially as you want to have more confirmatory outcomes, confirmatory tests, you're going to want to think very carefully about your analysis approach, your sample size, and your designs. Another big thing is what you guys are doing, and there's a lot of other resources that are out there, which is education about it. What should we do, and how do we improve our replicability and reproducibility practices? And there's a lot of stuff online as well. These are just two resources that I'm quite aware of. And then the one that I was trying to illustrate a bit by using the cancer biology project as an example is transparency. So a big part of helping increase replicability, reproducibility, and thinking about it is adding as much details as you can. So transparency of process and sharing as much as you can instead of having that kind of closed. It's really hard to assess what the differences are between two experiments if not both of those are as transparent as is reasonably can be done. So if I don't know half of the techniques or half of the methods that were done in an original experiment and I replicate it, it's really hard for us to advance knowledge because now I'm not sure unless I get the same finding, I'm not sure what's the difference. So transparency will help all of us, share your work and show your work. That's kind of the main principles to science. And when we think about it, I'm going to kind of end here in two little quotes, is it's more than just sharing data, right? And data can be a constitute for figures, right? That we know that there's a lot more underneath that surface, which is, yeah, we probably heard about sharing metadata and data management plans, but there's so much more that we should be sharing. right? And it's a lot of these practices, which is all those versions that you used, all your code, your data dictionaries, your raw data, your notebooks, right? The protocols you're using, even the hypotheses that you're trying to test. I think all of this is really critical to share, because if you just share the data, that's actually not going to help in the end, because we're still lacking all the necessary information that's below the surface that's kind of hidden. And it really does help advance research, both for yourself and for the others. And so I'm going to end on two quotes that I think are, I try to think really hard. This is one from early 90s, and it's talks about reproducible research in very much from a computational sense, but if you rip out the red words and put in different ones, it applies to all research, in my opinion, right? Which is, when we write our articles, when we write our scientific papers, it really isn't the scholarship. It's advertisement. It's a postcard for what was really done. That to be reproducible, it's actually everything that's behind the scenes. It's all the code, all the raw data, all the methods, all the reagents that were used. That's actually the research. That's the scholarship and the path that it took you to get there. And so I think that's something to think very hard about reproducible research, and the computational sciences is a really good way to think about how we can approach it. And the last thing I'll add is that the main person who's probably going to benefit it is yourself. Usually when your future self will thank your prior self for being more transparent, and hopefully other researchers will as well, but I'm pretty sure the biggest benefactor is yourself. So that's, to me, one of the biggest reasons to really think about how to make your work more reproducible, and how to design the best experiments and get the most bang for buck out of it every single step of the way. All right, I'll stop there. I'll end right here. Some more information, including these slides, are posted online. So thank you again for your attention.